Thanks.
All right, wherever she might be. We are on 13, y'all. 13. Can you believe it? We are moving along.
And this is our last one of genetics.
Yep. You guys have a test this week, right? Great.
Good.
Unit 4. We are wrapping up unit 4. Oh, geez. Yeah. That's the one spot I can see. That's why the blinds are down. Because this time of year, when the time changes and the sun rotates around like that, it is so hard to see in here. And it gets hot and it's bright. And so I put that there to kind of put the blinds down to kind of help. But it looks like Darcy picked like the one spot I can't fix. So you got everything you need for today? Okay, so labs, don't forget, turn in labs and turn in homework should be online unless you couldn't get it to upload or whatever. Yeah, lab 12. This week's lab is going to be due, the one we're doing today is going to be due next week. And we're going to go over most of it in class. So you should be able, if you're good at keeping up, you should be able to keep up. All right, let me see if I have anything to hand back. I think you guys have a couple things here. This is literally just a few. Looked over your stuff, Lil, that you had. It won't be right for long. It's going to move around. So the next thing happens this time of year. Oh, I'm sorry. I handed dude sitting on the couch. Sorry about that. The neat thing happens this time of year when the time changes and all. Somewhere around, usually around 10:30, when the sun gets over here, I have this little ball in my center of my table and my little centerpiece, and it's got like cracked pieces of mirror on it. It's just a little decorative thing, but the sun hits it and then it makes like a disco ball effect on the ceiling. It's really neat. It only happens for a few weeks out of the year. It's just the angles.
And every single day of the year, it like does little rainbow passes on the
Oh, little prisms. Yeah.
We have a chandelier in our entry room, and
Oh, yeah.
at like 3 o'clock to like 3-5 every single day, I'll hit it directly. And just, it's super pretty.
Yep. So this is usually like 10:30ish. So you'll probably see it. And then it's the same time of year that the sun is just at the right angle that it comes through. I have one of those frameless shower doors, you know, it's just the glass, and it hits the edge of my shower, like where the corner of my shower is, and it puts a giant rainbow right where I'm trying to look in the mirror, which is kind of, I mean, it's a huge rainbow. It's like this big, which makes it kind of hard to see what I'm doing, but it's cool. Awesome. The what? Oh, okay, cool. All right. Let me look at one quick thing and get this up here for you. Because it's not quite there yet, is it? I didn't send it. Go on. I didn't get it up there. Everyone had a nice weekend?
Yep.
Good weekend.
Good deal.
Unsurprisingly, the sun's been in my eyes the entire time. I sit at a desk over there, Lisa sit at a desk over there, the window there, right in my eye.
Oh my goodness.
I think the closest to the window, but Darcy's still gets the light.
You still get it, huh? It follows you around. That's crazy. There we go.
I don't think we want to do this.
Yeah, it's just so bright. We went up to the mountains. Drew had something going on in the mountains this weekend. So we went up to go pick him up, and gosh, it's pretty up there right now with all the colors changing and all.
So you like those yellowy, red, green, with the green leaves at the bottom and the red ones on top?
You know where I want to go? Has anybody been to Gibbs Gardens? I really, really want to go to Gibbs Gardens. It's a botanical garden up north towards the mountains, and it's really, really pretty. And it has a Japanese maple garden that's supposed to be gorgeous in the fall. And somebody posted pictures online of it yesterday, and I thought, oh yeah, I'm going. I gotta go. I'm gonna let this little
I tried not walking up Kennesaw Mountain.
He's got a new bone, so he's probably gonna go looking for it because he doesn't have it.
Also, Mount Neona. I also advise not walking.
Mount Yona? Yeah.
I walked up there once.
Oh yeah, we've done it. It's a long hike. Yep.
When I reached the gravel part, I was like, "We're there!" I ran up it.
Oh yeah. Did you see the big climbing wall on the side when you went up? Did you go up that path? That's where the Army Rangers practice.
I just went straight past him. It's great. He was like,
Yep.
"My bones there," it's like-
Yeah, he's got this new bone, so he's gonna be looking for it. All right, so we're starting our new lesson on genetics and genetic engineering today, but this is our last of the genetics unit 4. And next we're moving into concepts of changes over time. So we will, Anthony, question, is this yours? Is it? Okay, good. Because I asked like the last 2 weeks, but I think you were late getting in, so I didn't know whose it was. So I haven't credited you yet. All right, I'll make sure that I'll get you credit for it. All right. So this is some interesting stuff today. It should be a fun lesson. So genetic engineering. So this was a fun little picture. So by cloning cells and modifying genes, scientists in Korea have developed cats that glow bright red in the dark. And the cloned Turkish Angola, on the left, has a fluorescent protein in its skin cells. The protein gives off a red glow when exposed to ultraviolet light. The ordinary Turkish Angola on the right lacks the red fluorescent protein, so it appears green under ultraviolet light. Isn't that fun?
Is this like legal to do?
So we actually are going to talk a little bit about what's legal today. We're going to get a little bit into the ethics.
Depends what kind of fluorescent that is.
So we're going to get a little into the topic of ethics and legality
The
today.
cat looks radioactive.
It's weird, isn't it?
If it's radium, then that's gonna know.
But no, it's not radium because it's a gene. It's a modified gene. They take a gene from one animal, actually. So this is a transgenic animal. So they're going to take a gene from one organism and put it into the genome of another. And it's going to cause a production of a protein that's not typical. Because there are some animals that are bioluminescent, right? What's an example of an animal that lights up?
Some sort of Asian krill. But I was also gonna say that they put starfish DNA in the medicine you take for regrowing bones because they can regrow like entire portions of their.
There's all kinds of. Yeah, we're going to talk a little bit about that today too.
With jellyfish and certain snakes.
Jellyfish. Oh, really? Some snakes? Yeah. So jellies. Some jellies are bioluminescent. Fireflies, lightning bugs, as we call them in the South. Yeah. And then there are also some. There are some there's some mushrooms too that are bioluminescent.
What about those fish from the deep zone?
Yeah. There's some of those too, like the anglerfish with the little light in front.
I love the
There was this radioactive explosion in this town, and Lewis watches this thing called river monsters, and you just type on JrLRCL. They're like whales, catfish, I think, or whales. Was it? I'm not sure. Anyway,
I've seen that show. It's cool.
They mutate. I had nightmares for weeks.
Yeah.
They mutated, and now there's like rumored giant, huge mutated ones.
There are rumors about giant catfish in Lake Lanier that are not mutated. They're just rumored to be catfish the size of 12-year-old boys. And, there's all kinds of weird stories about Lake Lanier.
I'm scared of catfish because of that show.
So first we're going to talk about selective breeding. So this is exactly what it sounds like. So if I asked you to select your favorite candy bar from a big group on a table, what would that mean? What would it mean?
Twix.
Me too. Yay. Yeah. So you would pick out the one you want, right? You would select it. So that's exactly what this sounds like. So selective breeding is breeding organisms with desired characteristics to produce the next generation. So examples of how humans have done this is with horses, cows, dogs, plants. We do it all the time. We've been doing this for millennia. This is just looking at what you want and breeding to get what you want. So when you're talking about selective breeding, it's about allowing only those animals, or it should say plants, organisms in general here, with wanted characteristics to produce the next generation. So if you had, say you had a couple strawberry plants that just produced no strawberries, and then you had a bunch that produced tons of strawberries, you would take the ones that were non-productive and get them away, and you wouldn't try and replant them and breed them again. You wouldn't want those, right? They're not helpful.
Are those peppers?
Those are peppers. Yeah. So this is like how they wanted an orangey pepper. So they put this is like a codominance thing, I guess, where you could maybe put a yellow and a red together. And you do this in your own garden. I mean, people do this in their own garden. We only select the breeds of plants we know we're going to grow best, right? I mean, have you ever, you have a favorite brand? Some people have a favorite brand. They like Bonnie, or they like Burpee, or they like what, Monrovia. Those are all, yeah, that's a brand of vegetable, Burpee. Yep. So people will pick whatever they think is going to grow best. So also, humans have used selective breeding, which takes advantage of naturally occurring genetic variation to pass wanted traits to the next generation of organisms. So if you're a farmer and you've got cows, you're going to only want to breed your cows that maybe are the ones that produce the most milk, or maybe have the most muscle tissue, depending on what you're using them for. There's does anybody know where the cemetery in Cumming is? The Swanee View Garden. It's the memorial park. There's no stones, just little flower vases. Yeah, okay. So if you've ever been to the back of that cemetery, there's a bunch of cows that wander around. They're black cows. Well, no, but it's interesting. So I met the lady once that owns those cows, and they're called limousine cows. I didn't know this before, but limousine cows are cows that they specifically breed for beef cattle. So those are the mama cows. So they breed those mama cows, and their babies are for meat. But those cows, the mama cows, don't get eaten. They just breed again and again, because apparently they are the ones that produce the kind of beef people want. So this is what selective breeding is. And then also it produces new varieties sometimes, cultivated plants, domestic animals, horses, cows, cats, all this stuff, right? By selectively breeding for particular traits. All right, so colored cauliflower. Anybody ever seen the purple cauliflower or the orange cauliflower? I've seen it. Yeah, it's cool, right? So colored cauliflower is resulted from years of selective breeding. That's how we got it. So it has higher levels of vitamins than white cauliflower, and it's really pretty. So humans have also selectively bred wolves to create over 197 breeds. And there are 7 groups recognized by the AKC. Anybody ever watch the AKC dog shows? These are the 7 groups we recognize: toy hound, working, herding, sporting terrier, and non-sporting. Yeah.
I was about to say that on the internet, I saw a picture of this lady who had 2 dire wolves as pets.
Oh man.
And she used kids and like ride them.
I had a neighbor that had a dog who was like a quarter wolf, and he was freaking massive. His name was Lobo, which is Spanish for wolf, so it was the appropriate name. But he would stare through he had a 6-foot fence, and the dog was like this tall, but he would stare through the fence at you and just stare. And if you said, "Hey, Lobo," he'd start wagging his tail. But if you didn't call his name, he just stared, and he was really scary looking because he was so big. So who has a dog? I have a dog. Who has a dog?
My grandpa has a dog.
Okay. Anybody's dog. Okay. So what kind of dog do you guys have?
Mini golden doodle, and it's a weakling.
So a mini golden doodle is not recognized by the AKC. They call those designer breeds, right? Because basically they're made from 2 kinds of recognized breeds. What 2 breeds compose a golden doodle? Do you know?
Ours is first gen, so it's a mini poodle and a retriever.
Right. So golden retrievers and poodles. So golden retrievers would be in a sporting group. And I think poodles are as well. They might be non-sporting. Toy poodles are in the toy group, obviously. But poodles they were actually bred to do work in water, which is why you ever see the funny cut? You see a poodle and it looks like a French poodle, and it's cut with like tufts on its hips and puffy feet. It's to keep their joints warm. That's what that cut was made for. What kind of dog do you have, Anthony?
It's a mixture of a toy doodle and a golden doodle.
Okay, yeah. So you have the same thing. So designer breeds. So what's Ozzy? What kind of dog is Ozzy? He's a dachshund. And you know what group a dachshund would fit in? What would you think? Would it be toy, hound, working, herding, sporting, terrier, non-sporting?
Toy herding terrier.
Toy, herding, terrier. So it's actually hound. Yeah. So he's in the same group as things like bloodhounds.
Stash hound.
Yeah, it actually means badger dog, literally translated, right? So even though he's very small, he's actually bred for go to ground work, which is hunting badgers and rabbits that live in burrows. And you can usually tell hounds, there's 2 types of hounds. There's sight hounds and scent hounds. Sight hounds are the ones, you guys listening? They're the ones we've bred to see things really well, like greyhounds, whippets. These are dogs that run really fast, or borzois. They're really tall. Irish deer hounds, Scottish deer hounds. They've got great vision. They can see really far. Scent hounds are usually the ones with a long snout and big long ears, because that traps the scent. So,
it's kind of weird because our dog can run 35 miles an hour.
That's nutty. So you ever heard of a zonkey?
Yes.
So a zonkey is a hybrid between a
Oh
what?
my gosh, what is this new thing I am suddenly seeing? I want one.
What is it? It's a hybrid made from what?
It's a donkey and a
What is
zebra.
that?
A donkey
Donkey's a stupid name.
and a zebra. Okay,
Look
so
at a donkey and a parrot. Look at its like.
so
got,
it's
like,
so
parrot
listen, it's not,
feathers.
shh, don't yell, please. It's not a species of its own, because in order to be a species, it has to be able to reproduce, and like mules and things, they can't. But what can you tell about a donkey and a zebra if they're able to produce something? What must be true about their chromosomes?
I see their stripes.
Yeah. What must be true about their chromosomes?
That
They're
they've got
white.
like half zebra chromosome and half donkey.
So what does that tell you about the number?
There's 2 times the amount, I think.
If a zebra had 10 chromosomes, how many would you think a donkey would have?
20.
No, it would have to have the same amount in order to make an organism. So you know their chromosome number is the same, and their genes line up similarly. So what does a zonkey sound like? Let's see if my sound is at a good volume. I don't know.
Oh,
Here he goes.
turn it off. It's like that weird squeaky noise the zebra makes.
He's so cute though, look. She's talking to him and he'll talk back.
It's like you're cutting a saw.
It sounds like a killer.
He's so cute though. Look how cute he is.
He's cutting.
My
It's
dog
a
doesn't
saw.
react, he's like,
It's literally the sound of the saw.
I think he's cute.
I don't like it.
So hybridization is the crossing of dissimilar individuals to bring together the best of both organisms. And hybrid individuals are produced by crosses. Now, these crosses can take place, as I'll say in a sec. Oh, they can be hardier than both parents. And the crosses can be similar species, like in this case, or they can be varied members of the same species. So when you're talking about golden doodles, that's what you're talking about. It's a hybrid, it's a cross of varied members of the same species, because all dogs are the same species, but then we have breeds within a species, right? Like we've specialized breeds within a species. So you're crossing individuals that have very specific characteristics each and bringing them together. So if you have a look here, we're going to have some examples. So crossing dissimilar individuals to bring the best of both worlds together. So here's an example, a Brangus cattle. All right, that's an Angus cow. This is a Brahma. This is a Brahma cow.
Ugh,
Okay.
That's a bison.
So Brahma
What is that?
cows are
I
in
can't
India.
tell if I like it or if I hate it.
I hate it.
Does it grow its children from its shoulders?
Well, this is a male. Anyway, no, but
That's not helpful.
so they're from India and they can withstand heat. And Angus cows have really good meat, but when you put them together, you get what they call a Brangus cattle. So now you have a cow that has really good meat and can withstand the heat. So that can be advantageous.
Yeah.
So look, what do we have here?
Okay.
You're gonna get a golden doodle in this case, which is what you guys have.
That's what my dog looked like, only the poodle was not gray. It was like a dark
Uh-huh.
red.
Yeah. So they're known for being adorable and hypoallergenic. So the reason people started making golden doodles is because, so golden retrievers, dog trainers sometimes refer to them as lazy man's dog because they are very, very, very willing to please. That's their temperament. They want to please. They aren't hard to train. So a lot of people like the temperament of a golden retriever, but they shed a lot and they're a lot of work and they're big. So poodles don't really shed. They have like hair, not fur, and they're hypoallergenic. So if you have dog allergies and don't want hair everywhere, a poodle's a good dog. But people are like, this thing's kind of goofy looking, some people think. So when you put them together, you get this cute
My
thing.
mom friend has like a huge dog, a
Mm-hmm.
doodle that it's like a golden doodle, but
Yeah.
he's white.
Oh yeah, yeah. They have all kinds of variety. So what happens if you put these two together? What do you get?
Yes. I know.
A liger.
A liger, correct. If you put a tiger and a lion together, you actually get a liger. And
Is it like-
the
what
golden
if
doodle,
come up
wait a
with
minute.
names?
Shh. The golden doodle was known for being adorable and hypoallergenic. What's a liger known for?
Ferocious and
Like,
No, what's a liger known for?
come on.
Liger is pretty much,
Magical power.
come on, known for its skills and magic. Have you not seen
No.
Napoleon Dynamite? This
Wait,
is
okay.
homework.
Wait a minute. Raise your hand if you've seen Napoleon Dynamite.
Oh my gosh. Oh,
What
guys.
is wrong with it?
Shh, dude, don't yell.
Is it PG-13 because I'm allowed to see?
I don't even think it's 13. I don't know. It's the best movie ever.
Okay, I'm asking my mom.
You have to watch Napoleon Dynamite. It's hilarious. So anyway, pretty much my favorite animal, and it's known for its skills and magic.
It's probably PG.
So
It's an older
inbreeding.
That
All right,
one
so
people
shh,
can
Drew.
So,
So hybridization, you were taking organisms that were different and combining them to try and get the best. In inbreeding, you're taking organisms that are similar and trying to continue those
Wait,
characteristics.
Are they combined as one?
No, they're just holding on to each other. So there are problems with inbreeding. So continuous breeding of individuals with similar characteristics is inbreeding, and it can create serious problems. Used to maintain desirable characteristics in a line of organisms, helps ensure that characteristics that make each breed unique are preserved, and it can be risky. And this is why, because most members of a breed or a species are a breed within a species are genetically similar. So it's increasing the chance that a cross between two individuals will bring together two recessive alleles, and that can cause a genetic defect. Yes.
What's wrong with their eyes?
So this guy here has some degree of albinism,
That's a
probably.
woman.
But if you look closely, see these eyes? Now there's a couple ways that happens. That's called heterochromia, and that can happen in people too.
That's a woman.
And it could be, I don't really know. But there are a couple ways that happens. Sometimes it has to do with albinism, and sometimes it has to do with chimerism, which is like a very complex genetic thing. But if you ever see, sometimes you see animals in particular that have like one color face. Australian shepherds sometimes have a walleye or a watch eye, they call it. And those are merle. And actually, we're about to talk about the merle pattern. That's that patchy color that you get.
there's a hat that has like literally right down the center. This side's black and this side's white. This side has a green eye and this side has a blue eye.
So that cat is probably a chimera.
I love that cat. It's
And so that's a complicated genetic process. It's a little much to get into, but you could always look up chimera or chimerism. It's CH, spelled with a CH at the front. And it's interesting. It can produce some really unique looking organisms. All right, so we're going to take a second and we're going to talk about something I know a little bit about here, which is dachshunds. And the reason we're going to talk about dachshunds is because that's not why, but because there's a really great example within dachshund breeding. Yes.
Our dog's eyes are split up into 4 colors.
Oh, how pretty. That must be so pretty.
my friend's eye, like
Mhm.
1/4 of her left eye
Mhm.
is brown, but every other eye is blue, and the rest of that one eye is
Mhm.
blue too.
She
Cool.
has more than one eye. You said every other eye.
No.
I think she meant in her family. So we're going to talk for a minute about dachshunds because dachshund breeding gives a really, 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 really good example of the problems with inbreeding. All right, so dapple is a marking on a dachshund similar to merle. So when you were talking about Australian shepherds, you see the ones with the patchy colors, that's merle. In some breeds it's called merle, but in dachshunds it's called dappling, okay? And it's spotting, and it can be really, really beautiful. I've always wanted a dapple. I was going to look at a dapple female when we got Roscoe, our last dachshund. I had gone to look at her, but he won us over. So when one dapple dachshund is bred with another, you get a dog that they call a double dapple. So it can be really, really beautiful, but it has a very ugly side, this breeding process.
Oh,
So the dapple or merle gene for dachshunds is dominant. It's not recessive, it's dominant. And the combinations of the genes can be as follows. You can get, let me move this back up. So
wait, no, never
let me
mind.
move this out of the way. So you can get
Me too.
big M, little m, okay? And that's going to result in a dapple dog because the merle is dominant. This is a dapple.
I don't like the brown one.
You see the patterns?
Yeah.
Now here's the thing about dachshunds. Dachshunds for a breed have more variety in color, pattern, and coat than just about any other breed because there are 3 types of coats. There's this one smooth, see? These are both long hair, and there's also a wire hair. And then there's about 6 different color patterns that can go on those different coats. So dachshunds can have a lot of variety. So you got MM, which can result in a dapple dog, right? You can have MM, which is little m, that's recessive, and that's going to give you a non-dapple dog. So this is a black and tan like Aussie. This is a coloring called wild boar, which is more common on wire. That's a wire hair. And this is actually shaded. It's either a red shaded or a cream shaded. It's hard to tell, but that's another color. But none of these have, you see how none of these are dapple? This is dapple. In this case, he has patches of color, but these are called points. And this one has patches of color, and they're also called points because they're in specific areas. Whereas these are just kind of scattered, right? But if you put M and M, big M together, two dapples together, what you get is a double dapple. Now look at those dogs, okay? You might look at this, like these guys, and go, oh my gosh, they're really pretty because they're really unique looking, right? You don't see a dog that looks like that or like that very often. This one's pretty, right? And this one's kind of strange looking. He's got no color in his face.
Okay, yeah.
So
Okay.
this half the face. Now there's also a piebald with dachshunds where you get kind of a spotty pattern, more similar to like a beagle. But if you see a half a face like that, predominantly white like this, or a whole white face or whole white chest and upper body like this, that's a bad sign. That's a sign that you've got two dominant alleles and you've got a double dapple. And here's why it's bad. So for some reason, when a two dominant MM, capital M, are combined, they result in abnormalities, which can include blindness in one or both eyes. And sometimes you get a white patch covering them, like on the eye. Born with only one or no eyes, born deaf in both ears. And these problems can be lethal if not drastically reduce the quality of life. It can be lethal to have no eyes. Think about it. You could, I mean, for a dog,
Gonna go banging off the wall.
you could get hurt, right?
I've actually been on a horse without
So wait a minute, hang on. So dachshunds are already susceptible to certain health problems. And they are exacerbated in double dapples, which means they're worse. So these are problems that dachshunds already are kind of, likely to have, but they get worse. So it's things like spinal problems, intervertebral disc disease. Their long body, short legs causes problems with their little gait, their funny little gait, right? That's what gives them their little walk, but it also causes problems with their spine. They can have kneecap displacement. That sounds terrible. Your kneecap moves out of place. Your kneecap shifts around. Brittle bones. Cushing syndrome, which is a problem with cortisone in your body. Your body doesn't take care of its own androgen hormones the way it's supposed to, and thyroid problems. So intentionally breeding double dapple dachshunds is considered disreputable, and unintentionally breeding them is considered irresponsible. So if you were a breeder, what would you do if you had two dogs and you wanted to know if they were safe to breed? What are some things you could do to figure out? Yes.
Call the animal control or the government or something.
Well, they don't know. How might you figure out if it's safe? If your dog has two alleles, MM, or if your dog's even carrying one M, how do you know? What would you do? The vet could maybe give you some advice. So I'll tell you what people do: genetic testing. There are two main ones. The biggest one's called Embark, and you take a swab of your dog's mouth, just like we do with 23andMe and Ancestry.com, and you send it away. They send it back, and they can tell you your dog's genetics, and they'll tell you whether or not your dog carries the dapple gene, so you don't breed it to another dog that carries the dapple gene. So that's a practical application. All right, so cloning. Dolly. All right, so this is awesome for me because this happened when I was in college getting my major in biology, in the right kind of classes for this to have been the perfect topic to discuss. So cloning, a clone is a genetically identical organism. So technically speaking, identical twins are clones of one another, right? Genetically identical organisms. So in 1952, the first animal was cloned, and it was a tadpole. So you can see how this would be easy to do, right? Because tadpole eggs develop outside of a body, right? In a dish, you can do it in a dish. It's not like you have to keep them, they're not mammals, right? But Scottish scientists were the first to clone a mammal. And this happened in 1996. So I was right smack in the middle of my college years. This was my
My life is a lie.
fall of my junior year, I think it was. So it was Dolly. So Dolly, this is Dolly in the picture, this little girl right here. This is Dolly. She ended up dying from lung cancer and she had arthritis and she was euthanized in 2003 because she was having so many problems. But since then, there have been cloned cows, pigs, mice, and cats.
Why would they do such a thing?
Well, I'll tell you why, because there's good uses for it. So here's how it worked. So the way the cloning worked is, guys, there's a lot of side chatter today. The way the cloning worked is they took a sheep. And what they did is they took a cell from the sheep's udder. It was just an udder cell, the udders what the babies get their milk from. They took a cell from the udder and they got an egg and they implanted the information from the udder cell into an egg cell. And then they fertilized that. It was basically fertilized at that point because it had a complete amount of genetic information. And then they put that egg cell inside of another sheep who grew it inside of her and gave birth to this little clone named Dolly. Okay, so it was a real big deal when it happened. So you obtain the egg cell, you remove the nucleus, you obtain a somatic cell from the mother, which in this case was an udder, and you place the somatic cell into an empty egg. So they'd removed all the information from the egg. You grow it into an embryo, and then you implant it into a surrogate mother. What's a surrogate mother?
Like she gets her eggs.
Sort of. Basically, she rent. Humans kind of rent it because you get paid. But you allow the use of your uterus for the growing of an organism that isn't biologically yours. So in this case, this embryo was implanted into a sheep's uterus, and it wasn't her offspring. Humans do this too sometimes. There are sometimes human females that can't carry a pregnancy. So there will be another woman who volunteers to carry someone else's baby and deliver it. Actually, I knew a lady who was a surrogate. She was on her third pregnancy when I knew her third pregnancy as a surrogate, I was like, wow. But they do, humans get paid for that. It's a kind of a big deal to do, but yeah, unless it's a family member. Family members will do it for free sometimes. So genetic identity is equal to that of the somatic donor cell. So the baby was identical to the cell they got from the udder. So there are issues with cloning, obviously. So animal clones show high rates of death, deformity, disability. Dolly was the only success out of 276 tries, which means 276 either produced or potentially produced organisms were lost in the process. That's a lot. Is it natural? Well, would you want to be a clone?
No.
Is it natural?
It's kind of be cool if someone was like,
Yeah, is cloning a natural process? What do you think?
No, that is not natural.
Only if it's identical twins, I would say. Then otherwise it's a human-driven process. So would you want to be a clone?
No.
No?
So does that mean that you have duplicate yourself and all these stuff to it?
I mean, have they seen
Not
the
stuck to you,
movies?
no. It would be different organism entirely.
Have you seen the movies
Mhm.
where the clones turn evil?
Yeah.
I never see it coming. I hope for
So,
everyone's sake, no one invents
These are the ethical questions. So is it natural? Would you want to be a clone? Here's another one. Is it okay to clone human embryos for research?
No.
And then is it okay to destroy them if you don't use them? These are all questions that are part of the ethical process. Is it okay to clone human embryos for research and destroy them after? So sometimes they'll clone human embryos to grow specific tissues. And then when they no longer need that embryo, they'll destroy the embryo.
That's illegal.
Actually. So here's one though. How about cloning your own cells or those of a family member to replace damaged organs or tissues?
Now that's better.
Now so this is where cord blood banking comes in. Have you heard of this? So umbilical cord blood actually has stem cells in it. So if you, like when a baby's born, the umbilical cord is still attached to the mother and the baby, and they can draw blood out of that cord, and they can basically cryo freeze it and hang on to it, and you can pay for it. It's banking. It's called banking. You pay to keep the blood in a bank somewhere, in an ice cryo bank somewhere. And the thought is that as science continues to evolve down the line, we'll know how to take those cells and use them for exactly this kind of thing. Say you were in a terrible car accident and you got all your skin, a lot of skin burned off. They can take grafts and stuff, but that means they have to take skin from other parts of your body and move it around. What if you could grow your own skin that you got from stem cells from your cord blood when you were born? Or maybe not your own, but a sibling. So it's genetically close enough that your body's not going to reject it. So a lot of people are starting to do that, are starting to bank cord blood. They're keeping it. Actually, my doctor has his side business is cord blood banking. He has a whole registry where he's got cord blood bank. So I mean, it's a thing people are definitely doing. And I think within your lifetime, you are going to see the ability to do this. I think by the time, maybe not by the time y'all are having kids, maybe by the time y'all are having kids, or by the time your kids are having kids, it will be almost a common practice that you would want to bank stem cells, because they'll probably get so good, hopefully get so good at using them to replace your own stuff, which would be outstanding. Because think about it, if you have a injury or a disease and it damages something, do you know there's a type of diabetes that's caused by a virus? You catch a virus, now you're diabetic. So what if you could replace your pancreas with a new pancreas that isn't affected? What if, yeah, I mean, think of all the things that this could do. What if you had an eye injury and you could replace your cornea or your retina or whatever was damaged?
But then again, you
right?
have the risk of all that
Something
stuff.
going
That's
wrong.
true, but these are your own cells. So if they were, and you're not growing, in this case, you wouldn't be growing an entire organism. You would just be growing, they would be able to take the stem cells and differentiate them to whatever they want. So they wouldn't have to grow a whole organism. They would just grow pancreas, or they would just grow skin. And they can actually 3D print skin now.
Oh, that's
And some organs.
Oh,
It's wild. No, they can actually use cultures and print tissue. It's getting really, yeah,
That's good.
it's getting really cool.
Good thing. That needs to
So
start.
here are some of these benefits that we're talking about.
That's a good thing.
So if we improve cloning, we could reproduce animals with specific benefits. We could mass produce those animals. We could repopulate endangered species. That would be a good thing.
Like those white rhino experiments?
Yeah. We could produce whole human organs from a couple of cells. I would love this option. This would be a really, really good option.
Also,
Stem cell research can be from cloned embryos, which is, here we get into the questions again, like, are we messing with embryos versus stem cells are different? Like if you're getting stem cells from cord blood and using it to grow an organism, it's not the same thing as creating an embryo to use, right? So for you had these questions in your, well, you all had those. I'll move on to this part because I'm watching time. So genetic engineering. This is human manipulation of an organism's DNA. All right, so here's some examples of genetic engineering. Recombinant DNA, which we'll talk about in a minute, cloning, which we just talked about some, and transgenic organisms. All right, so what's recombinant DNA? So this is cool. This is when you join together DNA from two different sources. So what you get is the ability to change the genetic makeup of something. So here's a quick video on that.
A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as ECO-R1. The enzyme has a precise shape that allows it to run along the groove of the double helix, scanning, in the case of ECO-R1, for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted. When it cuts, ECO-R1 leaves a sticky end. This helps the new gene to attach.
Sticky end is what it's actually called.
The joins are then stitched together by another enzyme called DNA ligase.
it's honestly look
The
like
genetically engineered bacteria is grown in a culture medium. Very quickly, large numbers of the bacteria can be produced, each with a copy of the inserted gene. The bacteria duly manufacture whatever protein the gene codes for, and so the desired product is made.
So what do we use this for? Great example. You know anybody who's diabetic?
My Nana?
Does she take insulin?
Yes.
Probably grown this way. That's how we do it. We've taken a gene to produce insulin, put it in the genetic code of a bacteria, and then the bacteria produce insulin for us. And then we use it for medication. So transgenic, check this out. This is when you take a useful gene from one organism and you put it in another. It's a type of recombinant DNA in particular. So this is like the cat, but in this case, it's a tobacco plant that they've made glow by putting in firefly genes. So bacteria used to produce many important substances, like we were just talking about, insulin. They also do growth hormone, clotting factors. And then we have genes to make better meat, resistance to disease, faster growing animals, and they're important to the food supply. So 81% of soybeans and 64% of cotton have been genetically modified. And then we have genes for disease resistance that produce their own insecticide. They're heat resistant. They resist weed killing chemicals. All this is important stuff. Now check this out. Hopefully this isn't too long. I'm watching our time.
From anti-aging therapy in mice,
Oops, sorry.
animal carrying human cells,
Wait, this is cool.
livestock, and building organs outside the body, genetic engineers are constantly searching for answers to many social challenges. From global issues to medicine, these guys put everything to the test. One of the biggest goals of genetic modification is to reduce world hunger by producing more food. These featherless chickens have been genetically engineered to keep them cool.
Since I'm a geneticist, I was looking for a genetic solution. That means instead of cooling the environment of chickens with feathers, is helping them feel cool by just removing the feathers.
Without feathers and scales on their feet, these birds can grow comfortably in hot countries like Nigeria and Indonesia. The hope is that once successfully bred, the healthy commercial-sized birds will help feed underdeveloped countries. To fight global warming, some turn to enviro-pigs. The industrial amount of waste from pigs is a serious environmental pollutant.
Manure has a very high level of phosphorus within it from normal diets through normal pigs. And that phosphorus usually is more than is required by crops produced on land fertilized with pig manure.
But the remarkable thing is that the enviro-pigs carry a man-made gene. One part comes from E. coli bacteria, which produces an enzyme that can break down phosphorus. And the other from mice produces a genetic switch that allows this special enzyme to be made in saliva. These two pieces of DNA were combined to form a new compound gene that makes their food more environmentally friendly. Medical researchers across the world are also turning to transgenic.
Yeah, they do methane.
If you look at the word transgene, it means that the gene was transferred, a foreign gene was transferred to the animal.
Having successfully used transgenics to insert a jellyfish gene into a rabbit, when charged with blue ultraviolet light, the skin and the eyes of these rabbits glow green. But why make animals that glow in the dark? The incandescent gene allows researchers to easily track the movement of cells. By tagging certain cells with the glowing jellyfish genes, scientists can track which cells go where after an organ transplant. The technology is helping to find treatment for problems like blindness and bone disease. Glow-in-the-dark rabbits are just a start to the medical challenge that genetic engineers have taken on. No one said it was easy being green.
That's interesting. So real quick, we're going to breeze through this last bit because we're almost out of time. So agriculture and industry, 93% of corn produced in the US is genetically modified. Genetically modified crops can be resistant to viruses, yield more from less land. There have been over 20 years research on GMOs, and this is they're non-toxic. Studies have shown that GMOs have no impact on fertility or reproduction. Okay, so GMOs are hotly contested. You ever hear about GMOs? People talk about, I'm only going to eat organic food. I'm not going to eat anything that's GMO or genetically modified. Most things are genetically not. Drew, we don't have time for opinions because we're moving quickly. But stop. Most things are genetically modified to some degree. That's what selective breeding is. That's a type of genetic modification we've been doing for thousands of years. Some of this is a little different though. So everything we eat, much of what we wear come from living organisms, and use of genetic engineering to try and improve products from plants. So we do. And ideally, genetic modification could lead to better, less expensive, more nutritious food, as well as less harmful manufacturing. So pesticide, you don't want to eat pesticide. So they have found ways to genetically engineer crops so that they don't need pesticide. They are something that the insects won't eat because of the way they've been engineered. So GM crops were introduced in 96, 2007, 92% of soybeans, 86% of cotton, and 80% of corn. So we talked about some bacterial genes. They produce a protein called Bt toxin, and basically this makes them, it kills insects that eat them, and it makes them non-palatable to insects. So they don't have to put pesticides on these crops because they contain a protein that is basically their own pesticide. So cows, 30% milk from cows is injected with hormones made by recombinant DNA because it increases milk production. Pigs produce more lean meat or high levels of healthy omega-3 acids. Salmon has a growth hormone that they grow more quickly. Now, spider genes into goats.
Ooh.
We have one more quick video before we run out of time, and then I want to leave up. Oh, and here's the thing. In 2008, the US government approved the sale of meat and milk from cloned animals. Hold on, because wait till we wrap up because we're almost out of time. There's one more video here I'm going to show you, and then I'm going to make sure that you have time to look at your test subjects, okay? Because they're going to be up here so you can write them down to know what to study. But this is pretty cool. Cute little goat.
I'm Danielle Gassride. I am a master's student at Utah State University, and I work in Dr. Randy Lewis's spider silk lab. We are trying to produce the proteins that spiders use to spin all of their fibers. This is the spider that we base all of our genetic sequences off of. It's called the Nephilah clavipes. It's also called the banana spider. You can't farm spiders, they're cannibalistic, and you cannot get actual protein from them. So what we're trying to do is produce all those types of silks in other hosts. Those hosts include alfalfa, bacteria, we use E. coli, goats, and silkworms. The goats are not affected adversely. They are genetically modified from the time of conception, basically. So they're modified in the embryo, and then everything after that just comes from breeding. So when we breed the goats, not every baby has the gene. The only difference in the goats is they produce a lot of proteins in their milk. These goats are special because they produce one extra protein basically in their milk. So yeah, you tasted it, you look at it, you look at them, they look exactly the same. Spider silk is biocompatible, which is one of its really cool properties. So potentially we could use these products for medical uses and replace things that are used now, make them better, make them stronger, make them easier on your body. We've also had interest in the textile industry and in adhesives, making eco-friendly adhesives that don't have harmful byproducts, that aren't adhesives that aren't petroleum based. That's kind of the great thing about this research, is the kind of opportunities are endless, because it has the potential to do so much good. It would be a shame to not harness the power of genetics.
So when they milk those sheep, they go through the process of isolating the protein, and then they can produce spider silk at way bigger quantities than they can for spiders. I'm going to link this one. This is about how biologics are made. And this right here, just to show you, anybody recognize any of these?
Yep.
Anybody recognize any of those drugs? These are biologics. Okay, this is a class of drugs that's relatively new. They're relatively new, and these are vastly improving people's quality of life. It's a biologic that has gotten me back on my feet with lupus. Like without it, I would have been, I was drowning, guys. I mean, it has been a life changer for me. They are super expensive, and the reason is because see how much research goes into this stuff? Like lots of research goes into this stuff, and for good reason. So biologics, I'll post that video so you can see it because it's pretty cool. It's a how it's made video. And then there's just a little bit here on gene therapy, and it's about how we can replace, sometimes they try to replace individual genes. And also we can use this same stuff for fingerprinting, which we did in lab. So the same basic technology. And I'll post this last one here, but this last one is, I'll post this in your, sorry, I'll post this as well for you guys. But this is the first time DNA fingerprinting was used in a criminal case. Yeah. And they've used it for all kinds of reasons. Like, for example, wildlife conservation identifies herds from which black market ivory was taken. I mean, they can use DNA for that. So genome, you guys know what a genome is because we've been over it 100 times. And I'm actually wearing the human genome today, if you didn't notice. So here are your test topics. I'm going to put this up. You guys go through, write these down, and I'll call everybody else in. We're going to start lab right at the counter. Oops, sorry. I'm gonna put this up so when, after you've copied it, come on over to the counter and we will start lab over there. All right. Okay, gang. All right, Sky. Catch you later. Oh, hang on, I'll be right back up. It'll be back up.